Today is my great pleasure to welcome my colleague and friend, Jagan Mupidi from NCI to give us a talk. Before the seminar, I'm just to give a brief introduction of Jagan. Jagan actually got his bachelor degree in University of Kansas, and then he continues to stay in the University of Kansas to pursue his MD PhD degree in School of Medicine. And then after which he got his MD PhD degree, and then he started to start his residency in Weill Cornell in New York. And then from 2009, he moved to UCSF, started his clinical fellow in medical oncology. And then he did a serious clinical work. But meanwhile, he actually really got interested in basic research. He was doing his postdoc training in Jason's sister's lab, working on the B cells from 2009. And then from 2017, he joined NCI in the branch of lymph node malignancy branch, start his own independent group. Jagan actually focused on the B-cell lymphoma, and then did a serious great job in Jason's lab, and continued to focus on the B-cell lymphoma when he started his independent group. He has a series different awards. I think the most impressive is he continued to get the Leukemia and Lymphoma Society Fellow Award from 2012 to 2015, and then 2015 to 2016, showing he's really indicated in this field. And then he got also NCI Clinical Investigator Award from 2015. Today, we're gonna hear his newest work of Fas-mediated suppression of germinal center-derived lymphoma. And then go ahead, Jagan.
Okay. Just like to thank Chuan for the opportunity to present our work today. That's been recently published in the Journal of Experimental Medicine. It's always a great opportunity for a young investigator to be able to present in these forums, and I really thank him for organizing. As Chuan mentioned, the primary focus of my lab is understanding how genetic events that occur in lymphoma lead to the pathogenesis of malignancies. And specifically, we're interested in understanding how frequently mutated genes in lymphoma affect the homeostasis of the germinal center. So as you all know, germinal centers form at the center of the B-cell follicle during immune responses following immunization, but also occur in gut-associated lymphoid tissue during homeostasis. And current models of just get a laser pointer. So current models of the germinal center reaction suggest that cells undergo iterative processes of selection followed by diversification. And so selection of high affinity B cells is thought to occur in the light zone, which is marked by the presence of follicular dendritic cells which house antigen for long periods of time. And B cells compete for antigen on these FDCs and then present that antigen to T follicular helper cells that are present in the light zone. And when B cells acquire sufficient antigen to present to T cells, there's thought to be positive selection of these cells that can occur, and these cells migrate to the dark zone. And in the dark zone, they undergo rounds of proliferation as well as somatic hypermutation. Some of these clones can return back to the light zone and then undergo this process again. And over time, this is thought to lead to the development of high affinity clones, which can become plasma cells or memory B cells. So germinal center B cells are amongst the most proliferative somatic cells in the body, but over time, the germinal center itself remains relatively stable in size. And the reason for this stability is due to high levels of ongoing cell death that occurs in the germinal center. And so apoptotic germinal center B cells can be found in both the dark zone and the light zone. However, recent work from Nussensweig and colleagues, as well as one of my former lab mates in the sister lab, Oliver Bernard, has shown that distinct mechanisms account for death in the dark zone. So in the dark zone, the apoptotic cells that can be found are thought to primarily undergo apoptosis due to the introduction of deleterious mutations in their B cell receptor that prevent its re-expression on the surface. However, this mechanism does not account for the cell death that's seen in the light zone. And current models of light zone death, or current thoughts about light zone death, is that this is just thought to be due to neglect. So one of the questions that we had was whether or not there were any active processes that could account for death of cells that occurs in the light zone. So for a variety of reasons, we became interested in the role of the death receptor Fas in germinal center homeostasis. And so as many of you know, Fas is one of the markers that's used to gate on germinal center B cells. And so it's one of the most, it's a cell type that most highly expresses Fas. And it's previously been shown by Royeski and Mack in 2008 that loss of Fas in B cells specifically can result in the development of systemic autoimmunity. However, many groups have published by focusing on antigen-specific germinal center B cells that many groups feel that Fas is not required for maintaining germinal center homeostasis in a cell-intrinsic manner, and instead these groups have proposed a model that Fas is only acting at late stages of the germinal center reaction to prevent the emergence of aberrant memory or autoreactive plasma cells. So, so we wanted to revisit this topic when I started my lab here. And so one of the basic techniques that we've used is using bone marrow chimeras to look at cell intrinsic properties of loss of various genes in germinal center homeostasis. And so to do that, we generally mix either wild type or, in this case, Fas deficient bone marrow that's marked with CD45.2. We mix that at a 15% to 85% ratio with wild type bone marrow that's congenically different and is heterozygous at the CD45 locus, and so it's CD45.1/2. We irradiate CD45.1 hosts, then we wait 7 weeks, and we immunize these animals subcutaneously with a potent T-dependent antigen sheep red blood cells. And we can look in a variety of tissues. So we can look in peripheral lymph nodes for the immunized germinal center, which are analyzed by many groups, but we can also look in mesenteric lymph nodes and Peyer's patches where these chronic gut-associated germinal center reactions occur. And so when we do this with Fas deficient bone marrow, and we gate on donor B cells, you can initially see that in the follicular B cells, which are CD38 and IgD high, we can see that the representation of CD45.2 single positive cells is around 15%, which is what our input frequency was. And when we gate on IgD low and CD38 low cells, and then we gate on GL7 high cells, we can see that those CD45.2 Fas deficient cells in the germinal center are now expanded and account for around 50 to 60% of the cells in the germinal center reaction. And so if we do this across a number of mice in the mesenteric lymph node, you can see that in our control chimeras, the fraction of cells that are in the follicular B cells or the germinal center B cells is very similar. But then in our Fas deficient chimeras, you can see that there's this relatively large expansion of cells that are Fas deficient in the germinal center that are arising from a minority of follicular B cells. So when we look across tissues and we can use a ratio of the percent of cells that are CD45.2 in the germinal center compared to the ratio of cells that are CD45.2 in the follicle, in their control chimeras, the ratio is around 1. And then we can see this relatively large expansion of cells in the germinal center of mesenteric lymph nodes. And we can see an expansion in immunized settings, but surprisingly, we don't see any expansion in the Peyer's patch. So we think that there are tissue-specific cues that are required for enabling this Fas-dependent selection that we think occurs in germinal center, and that this selection occurs both in peripheral lymph node and in mesenteric lymph node. And I'm not going to show data, but we think we're still working on the reason why in Peyer's patch there's not this dependency on Fas-mediated selection. So Fas deficient mice develop systemic autoimmunity, and Fas is expressed on a variety of cell types, not just germinal center B cells, and it's expressed on T cells. And some of the autoimmunity that these mice develop is thought to be dependent upon Fas expression on T cells. And so one possible concern about this particular experiment is that these Fas deficient T cells are somehow contributing to an environment that allows for these outgrowths to occur. And so we wanted to address this by using floxed allele of Fas. So we used a floxed allele of Fas that was originally made by Klaus Rajewski, and we crossed it to CRE, the CR2 CRE mice where CRE is expressed on mature B cells. And we used these mice to generate mixed bone marrow chimeras, and then again looked in mesenteric lymph node after 8 weeks after reconstitution. And so again, in this setting where Fas is only lost on mature B cells, we saw this relatively large cell intrinsic expansion of germinal center B cells in the mesenteric lymph node. So as I mentioned, previous work has shown that animals that are deficient for Fas in B cells develop a lymphoproliferative type disorder and systemic autoimmunity. And so one issue with this system is, one caveat could be that these germinal center expansions are occurring as a result of that systemic autoimmunity. And so we wanted to come up with a way to get around that using polyclonal cells. And so the system that we came up with was to transfer polyclonal splenocytes that were a mixture of either Fas deficient cells or wild type cells into mice that were transgenic for the MD4 BCR, which is a henaglysozyme specific B cell receptor, and 99.5% of the B cells in these mice express that B cell receptor. And so we've shown previously that when we transfer polyclonal cells into these mice, we can get these cells to preferentially enter germinal centers against antigens that are not related to henaglysozyme. And so in situations where we have gut-associated lymphoid tissue, we can transfer these polyclonal cells and they can enter germinal centers in mesenteric lymph nodes or Peyer's patches because the antigen that is causing these germinal centers to form is not henaglysozyme. So when we do that and we look 10 to 14 days after transfer in the mesenteric lymph node, you can again see that Fas deficient cells are out competing wild type cells in the germinal center. And so this really suggested to us that this was a cell intrinsic expansion that was not dependent upon the systemic autoimmunity that's present as a result of loss of Fas-mediated selection. So Fas has been shown in a variety of systems to occur, the expression of Fas has been shown to occur before cells enter the germinal center. And so we wanted to know whether or not the selection that was dependent upon Fas was dependent upon what was occurring in the germinal center or before cells entered the germinal center. And so we just wanted to verify in our system that Fas expression occurred prior to cells entering the germinal center. And so to do that, we took splenocytes that were labeled with cell trace violet. And then we again transferred them into the MD4 animals that are specific for hen glycozyme. And then we could look at cells these polyclonal cells that were dividing and differentiating into germinal center at early stages using dilution of cell trace violet. And so that's shown here, and we have cell trace violet on the x-axis and then a germinal center marker FMB1 on the y-axis, and you can see that you can see early divisions of cells that are not expressing germinal center markers, and we think that these are pre-germinal center B cells. And so we can look at Fas expression on these three different populations: these naive cells that have not diluted CTV, these pre-GC cells that are starting to dilute CTV but don't express germinal center markers, and then actual germinal center B cells. And so you can see here that Fas is starting to be expressed on pre-GC cells and then is more highly expressed in germinal center B cells. So we can do mixed transfers in this setting using Fas deficient bone marrow mixed with, or Fas deficient splenocytes mixed with CD45.1/2 splenocytes that are wild type. And you can see that Fas is indeed not expressed on Fas deficient cells in the pre-GC stage or the germinal center stage. And then in our CR2-CRE system, where Fas is deleted on mature B cells, you can see that there's loss of Fas at the pre-GC stage and a partial loss of Fas at the pre-GCC stage, and then complete loss of Fas in the germinal center. And then we also crossed our animals to AID-CRE, and AID-CRE is primarily expressed in germinal center, and then we can look at Fas expression on these three different populations, and you can see that Fas is not lost in the pre-GC cells in that with that CRE system. So then we can look at the relative contribution of Fas deficiency to each of these three populations: the naive cells, the pre-GC cells, and the germinal center cells. So at day 5 following transfer, we can see that there's a small expansion in the pre-GC stage, and then a slightly larger expansion in the germinal center. And then if we wait to day 10, that expansion increases in the germinal center. So again, suggesting that that primarily this selection occurs in the germinal center, and that counts for this increase over time. So we can do this same system with the AID-CRE animals, in which Fas is not lost at the pre-GC stage, but is lost at the germinal center stage. And again, we can see expansions in the germinal center, no expansion in the pre-GC stage, and this expansion increases over time. So we can also generate bone marrow chimeras with the AID-CRE mice. And again, in the absence of Fas, we see this large cell-intrinsic expansion in germinal center B cells in the mesenteric lymph node. In this setting, we also don't see an expansion in the Peyer's patch. So we see cell-intrinsic expansion. We think this expansion is occurring in the germinal center. And so what sort of mechanisms are accounting for that expansion in the germinal center? So initially, we wanted to look at proliferation of Fas deficient cells in the germinal center. And to do that, we labeled animals with BRDU IP for 30 minutes. And these are mixed chimeras. And when we do that, we can look at the fraction of cells that are labeled with BRDU. And in Fas mixed chimeras, what we see is that, in fact, there's decreased amounts of BRDU incorporation amongst Fas deficient cells. So increased proliferation of these germinal center B cells is not accounting for the expansion that we see, the large expansion of germinal center B cells that we see. So we found that so we can also gate on light zone and dark zone using flow cytometry, using the markers that were originally described by Victoria. And so dark zone cells are high for CXCR4 and low for CD86, and light zone cells are relatively low for CXCR4 and high for CD86. When we look at the representation of CD45.2 single positive cells in the light zone and the dark zone, there's always a slight increase in the amount of Fas deficient cells that are found in the light zone. And so that's shown across a number of mice here. And we always see a slight expansion in the light zone compared to the dark zone. So we wanted to ask whether or not there was deletion of cells in the light zone, but not the dark zone. So as I mentioned in my introduction, in the dark zone, we think that the mechanism that accounts for death is due to loss of the ability of cells to re-express B cell receptor, but it's not really known what the mechanism of deletion is in the light zone. And so when we looked at the fraction of cells that were undergoing death in tissues that were analyzed directly ex vivo, we could see that in wild type cells, there was always slightly increased amounts of caspase-3 activity compared to Fas deficient cells. So if we look across a number of mice in wild type mixed chimeras, we don't see any difference in caspase-3 in the light zone of mesenteric lymph nodes, but we see increased death amongst wild type cells in the setting when they're mixed with Fas deficient cells. Now, this mechanism is not at play in the dark zone. In fact, we see very similar amounts of caspase-3 in the dark zone of Fas deficient cells. And that's shown here across a number of mice where we don't see a statistically significant difference in caspase-3 activity in the dark zone of Fas deficient cells. So we think that Fas is acting to delete some cells in the light zone of germinal centers. And so we wanted to next ask what the cellular source of Fas ligand was in the germinal center. So in data that I don't, so if we look at bulk sorted populations of NK cells or CD8 T cells, those do express some level of Fas ligand in cells that are directly analyzed, that are sorted and then analyzed by qPCR. But neither of these cell types are present in the germinal center. And when we've done depletion experiments, we found that neither of these cell types could account for the outgrowth of Fas deficient cells. So one cell type that is present in the germinal center is T follicular helper cells, and those, those cells do not express Fas ligand when we bulk sort them compared to non-TFH. So we wanted to ask whether or not on a single cell basis, if we could see any subpopulation of T follicular helper cells that could express Fas ligand. And to do that, we reanalyzed TFH single cell RNA-seq data from Stephanie Eisenbarth's group at Yale. And what we found is that Fas ligand was expressed in a fraction of all TFH clusters that were seen. This is just showing that about 20% of the TFH had some detectable levels of TFH in single cell analyses. Now, if we did gene set enrichment studies of comparing Fas ligand positive cells to Fas ligand negative cells, what we found is that there was an enrichment of T cell receptor signaling gene expression sets in the Fas ligand positive cells, suggesting that Fas ligand is made by cells that have recently been involved with recognizing antigen on germinal center B cells. So we also wanted to know whether or not we could actually detect Fas ligand in situ. And so detection of Fas ligand by protein was not possible technically. And so we decided to look at Fas ligand expression in situ using RNA-scope technology. And so initially when we looked, so we did this using human tonsil sections. And so the germinal center is here, and the light zone is here. And you can see in this blown up section that we can start to see that there are some cells that do express some detectable amounts of Fas ligand. These cells are in a similar distribution to T follicular helper cells, which are marked by PD-1. And notably, there are very few regulatory T cells that express FOXP3 in the germinal center in tonsil. So we also were able to do co-staining for PD-1 in red and Fas ligand mRNA, and you can see that there's the vast majority of PD-1 of Fas ligand is expressed in PD-1 co-expressing. So in situ, we can see evidence of a Fas ligand cell that is expressed. That Fas ligand is expressed by T follicular helper cells. So we wanted to know if functionally Fas ligand expression on TFH was required to suppress germinal center outgrowths. And so to do this, we again used bone marrow chimeras in which we mixed a minority of cells with either wild type bone marrow or Fas ligand deficient bone marrow with bone marrow that was deficient for SAP. And so SAP deficient bone marrow cannot produce TFH. So in these chimeras that are Fas ligand, that are mixed with Fas ligand deficient, the only TFH that are present will be Fas ligand deficient. And so then we can look at whether or not there are increases in the amount of germinal center B cells in the mesenteric lymph node. And so in fact, when Fas ligand is entirely deficient on T follicular helper cells, what we see is that there's an increase in the total number of germinal center B cells in the mesenteric lymph node. And this is not the case in Payer's patch, where we don't think that Fas-mediated suppression of the germinal center occurred. So just to summarize this part, we think that TFH express Fas ligand and are present in the light zone and are the cellular source that accounts for death of cells in the light zone. So what about an immunized setting? So recent work from both Gabrielle Victoria as well as Garnett Kelso suggests that that early germinal centers can be seeded by many clones. And many of these, the majority of these BCRs, germinal center B cells that are recruited into early germinal centers do not express B cell receptors that recognize the immunizing antigen. And over time, these weakly binding or non-antigen binding BCRs are culled from the germinal center reaction. And one way in which they're culled is through these clonal bursts where BCRs acquire a high affinity mutation and then rapidly take over the germinal center. But low-grade selection can also occur, and it's not really clear how that low-grade selection where non-antigen binding or weakly antigen binding cells are counter-selected in the germinal center might occur other than competition. So we can see this increase over time when we immunize animals with NP CGG, and then we can look at the frequency of cells that bind NP in the germinal center over time in wild-type mice, and you can see that very few of these cells can demonstrably bind NP at early stages of the germinal center reaction, and that increases over time. And so when we look at mixed bone marrow chimeras that are deficient for Fas in mature B cells in immunized mice, we can look at follicular B cells, and the input frequency in these bone marrows is around 15%. And then we can look in cells that don't bind NP, and these are cells in the germinal center that are NP negative, compared to cells that do bind NP. And surprisingly, what we found is that amongst the cells that did bind NP, there was no enrichment for Fas deficient cells, but there was an enrichment for Fas deficient cells in the cells that did not bind NP. And so that's shown here in terms of a ratio of germinal center to follicular amongst non-NP binding cells versus NP binding cells. So we don't see a difference in the wild type chimeras, but in the Fas deficient chimeras, there's this marked expansion of cells in the non-NP binding cells, but Fas does not provide much of an advantage at all amongst NP binding cells. Now, one concern is that these cells are mixed, are immunized with NP-CGG, and we're only really analyzing cells for the ability to bind NP and not really the ability to bind to chicken gamma globulin, or which is the other antigen that's present. So, are these non-NP binding cells actually truly non-antigen binding cells? So, we decided to try to answer that in two different ways. And so the first way that we wanted to go about answering that is by immunizing animals with ova and then staining with ova-specific tetramers. And so if we look in ova binding cells, you can see again, there's no enrichment of Fas deficient cells amongst the ova binding cells, whereas the enrichment of Fas deficient cells is primarily amongst the non-ova binding cells, and that's shown across a number of mice here. Now, again, it's possible that these non-ova binding cells are just very weak BCRs that are not able to bind to our reagents, and so we wanted to come up with another system to be able to answer that question of whether or not these cells were truly non-antigen binding. So what we did was we crossed the HEL specific allele MD4 to Fas deficient mice, and then we took a mixture of hen-egg lysozyme specific B cell splenocytes that were either Fas deficient or wild type. We transferred them into wild type mice that are congenitally different, that were immunized with a non-hen-egg lysozyme antigen, in this case, sheep red blood cells. And then we re-immunized them the next day with sheep red blood cells. And so sheep red blood cells do not contain hen-egg lysozyme, and therefore, if these cells are able to enter and persist in the germinal center, these will be truly non-antigen binding. And so, indeed, we do see that that Fas deficient cells are able to persist in these germinal centers, or enter and persist in these germinal centers, whereas wild type cells are not able to enter and persist. So, so what we came up with was that our model is that Fas acts to counterselect as a low-grade selection mechanism that operates in the light zone to delete either non-antigen binding or weakly antigen binding cells that are initially recruited into early germinal centers. And that is one mechanism that allows for the antigen-specific cells to accumulate over time, in addition to the clonal bursts that have already been previously described. So we don't think that antigen binding cells are susceptible or as susceptible to Fas-mediated death. And we're still trying to understand the mechanism by which these cells become resistant to that. And so that's an ongoing piece of work. So I am in, my main clinical interest is in trying to understand how perturbations in germinal center selection or germinal center homeostasis might be involved in the development of lymphoma. And so we wanted to know whether or not Fas signaling played a role in germinal center derived tumors. So most mature B cell malignancies are thought to arise from cells that are either in the germinal center or recently passed through the germinal center. And the heterogeneity of these malignancies is thought to be related to differences in the cell of origin. So we see a great diversity of different types of malignancies with varied predicted cells of origin. So the most common B-cell malignancy is diffuse large B-cell lymphoma, or DLBCL. And it's an aggressive lymphoma that is often refractory to treatment. And it's thought to either be derived from germinal center B cells themselves or cells that have recently exited the germinal center. And histologically, there's not really great histological methods to look at heterogeneity within lymphoma, but work from Lou Stout and colleagues almost 20 years ago now identified two subgroups of diffuse large B-cell lymphoma on the basis of differences in gene expression profiling. And that really led to the model that these activated B-cell-like diffuse large B-cell lymphomas are derived from cells in the process of exiting the germinal center reaction, whereas the more frequent subtype, the germinal center B-cell-like diffuse large B-cell lymphoma, is derived from cells in the germinal center reaction. And these two subtypes have different clinical outcomes following chemotherapy, in which the GCB DLBCL subtype has a more favorable response to systemic chemotherapy. More recent work from Lou Stout, as well as Margaret Shipp, has identified different subclasses of these lymphomas on the basis of genetic events in these lymphomas. And the most germinal center-like genetic subclass is the so-called EZB subtype, which is enriched for mutations, gain-of-function mutations in EZH2, as well as translocations in BCL2. And these are divided into two different subgroups on the basis of whether or not there's a MYC gene expression signature. And so these subtypes that express, that have this MYC gene expression signature tend to do much more poorly than MYC negative cells. But even in spite of that, there are some MYC negative EZB subtype tumors that continue to do poorly and are refractory to chemotherapy. And so when I was in Jason Sister's lab, we characterized this tumor suppressor G-alpha-13 that is frequently lost in GCB DLBCL. And we found that in mice lacking G-alpha-13, that aged mice can develop tumors that originally arise in the mesenteric lymph node. And so one of these tumors is shown here in a knockout animal that was around 15 to 16 months old. And so when we analyzed these mice by FACS, what we found is that a high frequency of these tumors, in fact, had loss of surface expression of FAS. And about 40% of these G-alpha-13 deficient tumors had loss of FAS. And so what we did was we crossed G-alpha-13 deficient animals to mice lacking FAS, and we found that if we had, we generated mixed chimeras using these mice, and we found that animals lacking both FAS and G-alpha-13 showed synergistic effects on germinal center expansion in the mesenteric lymph node. And so we wanted to know whether or not FAS deficiency could play a role in the pathogenesis or clinical outcomes of human tumors. So if we look across the various gene expression subtypes, FAS is most highly mutated amongst GCB DLBCL. And when we looked in the more recently described genetic subtypes, FAS was most frequently lost by both mutation or deletion in the EZB subtype. And about 25% of these tumors had loss of FAS either by mutation or deletion. So these mutations primarily affected the death domain and led to truncation. And so these would be expected to act as dominant negatives even in heterozygous situations. And so more than 2/3 of these mutations were predicted to either disrupt or delete the presence of a death domain. And so when we looked in our cohorts of patients that were GCB DLBCL, and we look at Fas wild type cases compared to Fas mutant cases or Fas deleted cases, what we found is that there was a very poor survival amongst Fas altered germinal center B cell DLBCL. And this was not the case amongst the activated B cell like diffuse large B cell lymphoma. And when we looked in the EZB genetic subtype cases, what we found is that there was in fact still this poor outcome, and data that I don't have time to show you, this decreased survival was independent of the presence of a mixed signature. So when we looked at mutations that were co-occurring with Fas deficiency amongst these Fas mutant cases, what we found is that the vast majority of Fas mutant cases also harbored mutations in TNFR SF14, which encodes for HFEM. And a subset of these also were enriched for mutations in CD274, which encodes PDL1. And so recent work from Jason Sister and colleagues showed that HFEM acts as a negative regulator of T cell help. And it acts in the absence of HFEM. There was an expansion of weakly antigen binding cells in the GC, very similar to what we see in Fas deficient bone marrow chimeras. And then work from Hai Chi and colleagues also showed that PDL1 PDL1 loss on germinal center B cells was sufficient to allow for a similar expansion of weakly antigen binding germinal center B cells. And so when we look in these Fas deficient cases, what we see is that there's enrichment for TFH signatures, suggesting that these tumors are more dependent upon, as a result of the loss of these multiple negative regulators of T cell help, that they're more dependent upon the presence of TFH for their expansion and survival. So our current, the model that we're currently testing is whether or not loss of these various negative regulators of the germinal center TFH interaction can promote the development of malignancy. And so we're currently testing whether if we combine loss of Fas with loss of HFEM or loss of PDL1, that whether or not we can see increased development of tumors. And so with that, I'd just like to thank the primary people that did most of the initial mouse work. Were two post-bac students that were formerly in my lab, Rod Razagi as well as Shreya Agarwal. We had a great deal of help in the germinal center and the lymphoma studies from one of my mentors, Louis Stout, as well as Boston Gene. Some of the initial work with the G-alpha-13 deficient mice I did when I was a postdoc in Jason Sister's lab. And we had help with the single cell analysis from Chen Yao and John O'Shea's group. And then in work that I didn't have time to describe, we were able to look at Fas expression across a wide variety of germinal center tumors with the help of David Scott at the British Columbia Cancer Agency. And so with that, I'd like to take any questions.
Thank you, Jagan. That was a great talk. If anyone have questions, please type in the chat box, or you can just raise your hand. I think I'm going to start it. So I have a couple of things. One is actually relatively naive question. So I noticed that when you actually generate the bone marrow, you always use 15 versus 85. Mm-hmm. What's the reason behind that?
So we wanted to allow for sufficient dynamic range to be able to see an expansion. So in situations where we are predicting there to be an expansion, we always start off with a minority of cells that are lost for various genes. So if you it's just so that when we do the ratios, we can see more of a range.
So in that case, the outcome of the donor and then host are going to be still you you will still expect to be 1585?
Yes. Yeah.
Okay. The other thing I want to ask is, although you say there's no, you're still actually working on the reason why the Fas patch has no difference in terms of Fas deficiency. So I'm just curious that whether it's possible. Well, I mean, if it's possible, the reason of that consequence is because of the microbiome. But
yeah, so we think that it partially plays a role. So hold on one sec. So if we generate mixed bone marrow chimeras that are Fas deficient mixed with wild type, and then we treat them with antibiotics in their water for 7 weeks, what we can see is that in the control the normal chimeras, we don't see an outgrowth. But then when we deplete the microbiome, there does appear to be some Fas dependency. And so our current thinking is that there are microbially derived signals that are either acting on intermediates or acting on germinal center B cells themselves that somehow suppress the ability of Fas to signal for death. And so we don't really understand the nature of those signals yet, whether or not they're dependent upon either intermediaries or in the germinal center themselves. But that's definitely something that we think the microbiome is part of the reason that Fas-mediated selection is not occurring in the Peyer's patch.
But in that case, the only thing different from the Peyer's patch and then the mesenteric lymph nodes is the M cells, which actually express on the Peyer's patch, but not on the mesenteric.
Well, not necessarily. So as you know, the mesenteric lymph nodes drain a wide variety of the segments of the small intestine. And so the lamina propria drainage is going to the mesenteric lymph node, but not necessarily going to the Peyer's patch themselves. The anatomy is also quite different in the Peyer's patch, and I don't know if that can account for differences. So it's not just the M cells, but they're, the dendritic cells that are present in the Peyer's patch have very different functions compared to the mesenteric lymph node. For example, class switch to IgA occurs in the Peyer's patch much more highly than the mesenteric lymph node, and that's accounted for by the DCs in the Peyer's patch, but it's not at play in the mesenteric lymph node. So there are other differences that we need to tease apart that we still don't understand.
So in that case, if you use antibiotics treatment and then look back to the mesenteric lymph nodes, the difference before and then, on the Fas deficiency bone marrow, on the mesenteric lymph nodes, the difference will going to be even larger compared with the SPF condition.
Oh, in the mesenteric? Yeah. So it goes up a little bit too compared to SPF. Yeah.
Okay. So the microbiome does also affect the.
It affects the mesenteric. Yeah. Okay. It doesn't affect the peripheral lymph node though.
Okay. The other question I have is you were talking about the Fas ligand expressed on the TFH actually influenced the outcome of the B cell apoptosis.
Mm-hmm.
I guess what I want to ask is, can you comment on the other potential cellular source which can influence the outcome of the B cell apoptosis in additional to T cell, TFH cells, for example, like dendritic cells, things like
that. In terms of Fas ligand?
Yeah, in terms of Fas ligand.
So in situ, we only see, like, maybe there's a couple of spots of mRNA that are not co-localizing with PD-1. So I don't know if there's a substantial source there. We don't think that FDC express Fas ligand, at least based upon published single cell of FDC that's been done by both Jason's lab as well as Burkhard Ludwig's lab. So we don't think that FDC could be the cellular source. There aren't any dendritic cells, conventional dendritic cells that are present in the germinal center microenvironment. And whether or not there are other stromal cell types, I don't think that they're accounting for the primary effect. I think this is a TFH primarily.
So the single cell analysis or the single cell data which you analyze is purely on the T cell based, or
TFH, yeah.
Is there anybody look at the other cellular single cell based, let's say stromal cell, like you said?
Well, so the FDC have been analyzed by a number of groups, and I don't think that Fas ligand is prominently expressed on FDC, either the conventional FDC, which are light zone FDC, or the newly described dark zone FDC.
And also, if the TFH is the primary source of Fas ligand, do you see the spatial distribution difference? Let's say,
well, the TFH are primarily in the light zone, right?
Yeah, but I'm just saying that, I mean, the analyzed on the single cell basis including both dark zone and light zone, right?
So the TFH are not present in the dark zone, right? So
right. No, I'm just saying that the single cell analysis you actually look into is based on the entire.
It's on sorted TFH.
Yeah. Oh, okay. Yeah. Sorry. Okay. I see.
Yeah, but the other single cell is on stromal cells that are then processed for single cell. Yeah. So I don't think Fas ligand is prominently expressed.
Yeah. Got it. Okay.
And that includes both light zone FDC as well as the newly described dark zone FDC.
Yeah, but there's no.
I don't think so. I have to look back at the data, but I don't think.
Okay.
Prominent source.
Okay. Thanks. Any other questions?
Hey, Jagan. This is a really beautiful talk. So I have two questions. The first one is about the Fas. So how the distribution of the Fas expression look like on these like B cells in light zone? Is that a single population or multiple?
So Fas is expressed on both light zone and dark zone, and it's expressed at a uniform.
Pretty uniform. Okay.
Yeah,
I see.
Yeah, there's not a Fas positive or a Fas negative germinal center. Even if we use other markers to define germinal center.
I see. So it's a uniformly like a highly expressed.
Yes. Yeah.
I see.
So most groups that work on many groups that work on germinal center primarily use Fas high, CD38 low as the only markers that they use to define germinal center on FACS.
I see. So you mentioned there is like even like both these like BCR high and BCR low cells are expressing the Fas in the light zone, but they have like different differential like susceptibility to.
Yes. Yeah. So we don't see.
Yeah. Have you tested like other type of cell death, let's say pyroptosis, or like in addition to apoptosis?
So I don't know of any ways to look at pyroptosis by FACS on ex vivo samples. If we're trying to look for histologic evidence of pyroptosis, is that what you're thinking?
Well, I'm just thinking like if there's any like synergistic like mechanism to promote the cell death, like in addition to the Fas.
Oh, yeah. No, I think that's an excellent question. I don't know the answer to that. I don't think anybody has I can't think of anything that's specifically been published about pyroptosis in the germinal center. I don't know if pyroptosis would be advantageous to the germinal center reaction, just because it's, would be more inflammatory. Yeah, so, but that's something that we could definitely try to look at. I just don't know of a way to easily look at it.
Yeah, because I think that the differential susceptibility to the cell death, even you have the same Fas expression, that's very interesting.
Yeah, so we don't understand the mechanism by which that differential susceptibility occurs, and that's something that we're definitely interested in. We don't think it's related specifically to some of the downstream of the BCR, so we don't think that BCR-driven NF-kappa B is accounting for that difference. And we also don't think that CD40 ligand, CD40 interactions are accounting for the difference.
I see.
So whether or not there are other things that the T cell, there are definitely other things that the T cell could be providing to a positively selected germinal center B cell that could account for that, but we haven't dissected that in vivo yet.
Of course, the Fas and Fas ligand interaction, so that's like cell contact dependent, right?
So Fas is both secreted and expressed on membrane form, and there are tools to look at that we don't have. So there are mice that express only membrane form or only soluble form, and the mice that only express soluble Fas ligand do tend to develop autoimmunity, whereas the mice that only express membrane form do not. But whether or not that is due to a B cell level interaction or B cell killing hasn't really been investigated yet.
I see. So that's kind of suggesting the membrane bound fold is more potent in activating cell death. Is that?
Yeah. Right? So my thought would be that it is dependent upon a membrane, a cell-cell contact. So whether or not those are more short-lived, and that's why they account for the death in the weakly antigen binding cells, would be a possibility.
Yeah. So I think maybe it will be interesting to look into that, like if it's like the binding, the wild time will be like critical for the potency of these, right? Yeah, in vivo may be difficult. Well, still maybe can be done through like photon imaging of this kind.
Well, so I think one of the issues is that many of the 2 photon studies that looked at selection are using antigen specific transfers.
Yes.
So either whether or not those be B18 or like NP specific B cells or heterolysozyme specific B cells. And primarily the death that we're seeing is in the non-antigen binding fraction. And so I think having a 2 photon system to be able to look at that is going to be very technically challenging. Yeah.
Or just a simple in vitro system, like look at the cell-cell interaction.
So we've also had a lot of trouble with in vitro systems.
Okay.
So
it's a tough problem.
Yeah.
Cool. So I have another question, which might be like remotely related. So in metastatic cancer, so the like a nearby lymph node usually get enlarged. So how will that affect the germinal center like tissue homeostasis or like the development of these like?
So I don't actually know the answer to that, but I think that there would be suppression of just any type of reaction. I can't think of a study off the top of my head that looks at the looks at whether or not there are still germinal centers, that germinal centers can form in metastatic lymph nodes, but my prediction would be that it would be a very suppressive environment for the germinal center. Like there would just be high amounts of steroid or something like that. I don't actually know the answer. That is a good question, though.
I see. Well, I just wonder, like compared to the primary side for these like tumors, whether the germinal sensor or like these lysomes.
Well, sorry, I take it. So I'm just thinking there are these recent studies, maybe a couple of years now, looking at tertiary lymphoid structures being a marker of patients that do better. And so tertiary lymphoid structures are like isolated lymphoid follicles and that house B cells. Whether or not they house germinal center is another question. I don't think that they have looked, and those tend so there is some evidence that B cells can play a negative role. But
okay, that's interesting. Cause some of these like lung carcinoma, they also express like a higher like Fas as compared to the normal tissues.
Right.
I just wonder like if in the lymph node it provide a different like microenvironment as compared to the primary tissue.
Well, but I think the seeding would probably be in the T cell area, right? Whether or not the initial seeding, I don't think that you would expect there to be seeding
Okay.
in a B cell area. So in the T cell area, then I would expect there to be CD8s that would express
okay.
high amounts of Fas ligand.
Okay, great. Cool. Thanks so much.
All right, thank you for having me. I really appreciate it.
Anyone else has questions? If not, then thanks again for joining us. Got wonderful talk, and then really appreciate it. It's great.
All right,
so
thank you.
hopefully next time we can have you one more time. So I hope see everyone again this weekend. So thanks a lot, Jargon.
Thank you.
Yeah.